Hello, 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 hello. Hello, 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 hello. Hello, 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 hello. Hello. Can you hear me now? And it's not echoing? No? Okay, brilliant. So far away with the questions then. Otherwise, you're going to have to sit there and watch me draw for the next couple of minutes. Or if you like, I could pull up the exam questions that we did last week and carry on going through those. It's up to you. Structure of enzymes. Okay, Jag, no worries. Right then. So, let's have a look. Get rid of that, get rid of that. So, enzymes. Okay. So, if we start with a green keyword. So, an enzyme is defined as a biological catalyst, biological catalyst that's made of protein. Okay, so the important thing here is that it's made of protein. Okay, it's this idea that enzymes are made up of proteins. Now, when you did topic 2, you would have spoken about proteins having a primary structure. They have a secondary structure. They have a tertiary structure. And they have what's known as a quaternary structure. So quaternary. Okay. Essentially what this means is its first structure, its second structure, its third structure, and its quaternary structure, its fourth structure. Okay. So if we think about the primary structure of a protein, what we're saying is that it is a polymer or a chain of linked monomers. So it's a chain of linked single units that repeat themselves. The primary structure of a protein is a polymer of amino acids. Okay, so of amino acids. These are known as a polypeptide. Okay, a polypeptide. Now, the reason it's known as a polypeptide is because you've got these peptide bonds that join the amino acids together. So you've got a chain, a polymer of peptide bonds holding amino acids together. That's the primary structure of a protein. Okay? The secondary structure of a protein is when this chain of amino acids, so if we use blue as our chain, the primary structure will be a link of amino acids joined together like so. Okay? The secondary structure is when this polymer, this polypeptide, folds into either what's known as an alpha helix or a beta sheet. Okay? So the secondary structure is either what's known as an alpha helix or a beta sheet. Okay, a beta sheet. Now what this means is that the polypeptide folds round, in the case of an alpha helix, it folds round into a spiral shape. This is due to attractions between different parts of the amino acids. So instead of having a chain like this, in an alpha helix, what you'll have is sort of this helix shape, like DNA, like winding staircases that fold round. Okay. Another type that you can get is known as beta sheets. That's where it sort of becomes pleated. Yeah, it sort of folds on back on each other and becomes crumpled. Okay. Tertiary structures are when the secondary structure fold into a particular shape. Okay, so it's when the polypeptide folds into a shape. Now this is where it's important for enzymes. Okay, this is where we're concerned with enzymes. So if I pick, we'll have to use green because that's the only colour I've got. Tertiary structure is where it affects enzymes. Okay, in the tertiary structure of an enzyme, of a protein, what you have is these very important bonds. You get things known as disulfide bridges. You get hydrophobic interactions. You get hydrogen bonds, and you get other types of bonds. Okay, and what this does is it causes this folded shape to fold round even more. So for example, instead of being this straight curly line, what you'll get is the curly line folding round to form a particular shape. Okay, and it'll keep folding round like that. Now, in terms of enzymes, it's this tertiary structure that allows the active site to be made. Okay, so if this is the tertiary structure of a protein, this part may be the active site. And it's only if this tertiary structure is maintained that the active site keeps its shape. Okay, so if this is the active site, it's these disulfide bridges, these hydrophobic interactions, these hydrogen bonds that hold the enzyme in its particular shape, that allows the active site to keep its shape, and therefore the enzyme can do its job. Okay, if an enzyme gets denatured, what we get is these disulfide bridges or these hydrophobic interactions or these hydrogen bonds break, and that causes the tertiary structure of the protein to change. It causes it to fall apart. It's no longer folded together. If it's no longer folded together, what that means is that the enzyme no longer has an active site. It can no longer do its job. So denatured enzymes, so if we want to get really specific about it, denatured means when the tertiary, I do apologise about my handwriting, it will get better as we go. The tertiary structure of an enzyme changes. Okay, so if this tertiary structure changes, the enzyme no longer has its active site with its specific shape, therefore it cannot do its job. It cannot break down the substrate. Okay, so usually you have the enzyme and its substrate. The enzyme has this tertiary structure that gives it its active site. The substrate fits into the enzyme to form an enzyme substrate complex that breaks down the substrate and it makes products. Okay, quaternary structures of proteins, just to round this up, are made when you have more than one polypeptide chain wrapped around each other. Okay, so these are made when you have more than one polypeptide chain. Okay, does that answer your question, Jag? Does that help with the structure of enzymes? Okay, has anybody else got any other questions? Has anybody got anything you want me to go over? Anything at all? Is there anything people don't get? If there's something you don't understand what I've gone over, please just write in the chat so I can see. Meiosis and mitosis. Okay. Meiosis and mitosis. Let's just wipe the board. Don't want to save that. Change it to black so that we can actually see the board. So. Okay, meiosis and mitosis. So to begin with, cell division. Okay, cell division. Your body, it's said that after 5 to 7 years, I think it is, that you have no cells that are the same as when you first started. Every 7 years, I'm pretty certain it's 7 years, every 7 years, all of the cells of your body are replaced. They're replaced with new ones. Therefore, it's important that your cells can divide. It's important that they can make new cells. Otherwise, if that didn't happen, after 7 years, all your cells would die and there'd be nothing left of you. Okay? So, cell division is when cells divide to make copies. Okay, it's when cells divide to make copies of those cells or of other cells. Okay, now There's two main types of cell division. Okay, there's two types that are going on in your body right now. We have mitosis and we have meiosis. Okay, we have mitosis and we have meiosis. Now, the good news is for you guys is that because you're doing Edexcel and you're doing the Salters-Nuffield specification, they require that you know about mitosis and its stages. However, they do not require that you need to know about the stages of meiosis. You just need to be able to understand what it is and why it's important and how it's different from mitosis. Okay? So, let's just give a brief definition of the two before moving on to what they're used for. Okay? Let's look at mitosis first. So before we look at mitosis, we need another definition, which is somatic cell. Okay? Now, somatic cells are just body cells, okay? They can be your skin, they can be your teeth, hair, liver, muscle, bone, whatever, okay? So somatic cells are normal body cells, okay? And what's important about these normal somatic cells, these somatic cells of your body, is that they have a full set of DNA. Every somatic cell in your body contains a full set of DNA. There are 46 chromosomes arranged into 23 pairs. Okay, so because this is important, we'll switch back to red. So they're normal body cells, and they have which have a full set of chromosomes. So we can say another key word, which we won't write down, but we'll just put in brackets here, is that they are diploid. They are diploid. Okay? Diploid cells have a full set of chromosomes. Okay? Now, mitosis is the division of these somatic cells. Okay? It's the division of normal body cells. So, mitosis is defined as the division of somatic cells forming, and this is the important point now, so I'll go back to red, forming two identical daughter cells. Okay? Daughter cells. So in mitosis, you have somatic cells, which are normal body cells, which are diploid. They have a full set of chromosomes, which divide to form two identical daughter cells. The cells are identical. They are clones of each other. Okay? So mitosis, broadly speaking, is needed for, so it's needed for, if I can spell, it's needed for growth, repair, and also in some organisms it's needed for asexual reproduction. Asexual reproduction. Now, what asexual reproduction is, for those of you that don't know, is when you some organisms can reproduce, they can divide by simply breaking apart and growing into two new organisms, okay? And they do this through mitosis. There's no sperm or egg involved, there's no fertilization. So mitosis is the division of somatic cells forming two identical daughter cells, and it's needed for growth, repair, asexual reproduction, okay? Obviously, we cannot reproduce asexually. If we could reproduce asexually, I'd produce another Mr. Abraham who'd be out in the garden now enjoying the sun, and there'd be another one in here working with you guys on YouTube. Now, as I said at the beginning, mitosis for Edexcel, you need to know the stages, you need to know what happens in each stage, and you need to know them in order, okay? Now, when I was at university, I had a very good tutor who would be like your form tutor, but when you guys go to uni, you'll see that you also get a tutor. And he taught me a brilliant way of remembering these stages, okay? The way he said it to me was to remember, I proudly miss all tutorials. And considering you're in year 13, this is very, very applicable to you guys, because my year 13s, I'm lucky if I see them in a week, let alone in a day. So I proudly miss all tutorials. These are the stages of mitosis. They stand for interphase, prophase, metaphase, anaphase, and telophase. Okay? Those are the stages of mitosis. Now let me just quickly check the live chat to make sure we're still there. Yes, we are. So, I proudly miss all tutorials. Interphase, prophase, metaphase, anaphase, and telophase. Okay? As I said, for mitosis, you guys need to know the stages. Okay? Now, if you just give me 2 seconds, there's somebody at the door, so just give me 1 second, please. Right, and I'm back. So, mitosis. I proudly miss all tutorials. I'm afraid for the case of Edexcel, you need to know what these stages are. So if we quickly go through them. Right then. So, to begin with, first stage is interphase. Okay? Interphase. These are the stages of mitosis. Remember, mitosis is the division of somatic cells, and it results in 2 identical daughter cells. Okay? So in interphase, this is where the cell is sort of in its normal resting phase. Okay? So this is where the cell is in normal resting phase. This is essentially when all of the DNA of the cell is doubled. This is where DNA replication takes place and the cell grows. Okay, so in interphase, DNA replication happens. Okay, this is where DNA replication happens. So the DNA is doubled and the cell is in resting phase. Okay, You also get things like organelles that are doubled, the cell grows, as I mentioned, and you also get ATP being produced because mitosis takes a lot of energy. Therefore, ATP needs to be produced in interphase to get the cell ready to undergo the other stages. Okay? So, in this stage, the cell is just in its resting phase. Its DNA in its nucleus is still all stringy, like so, and it's just growing, it's just getting ready for the next stage. So if we follow the I proudly miss all tutorials, the next stage of mitosis is prophase. Okay, prophase. Now, prophase is where the chromosomes of the cell become visible. So in interphase, the DNA is all stringy, and then in prophase, the DNA condenses and forms these chromosomes. Okay, so chromosomes become visible. Okay, so the DNA condenses to form chromosomes. What we mean by condenses is they get shorter and shorter and fatter so that we can see them. Okay. Also in prophase, we get these organelles known as centrioles that move to opposite ends of the cell. Okay, so centrioles move to opposite ends of cell, and we also get the nuclear membrane breaking down. Okay, so the nuclear membrane breaks down. So we've had interphase where the cell's normal, it's grown, it's doubled its DNA, it's got more ATP. The cell then goes into prophase where the chromosomes become more visible, centrioles move to opposite ends of the cell, and the nuclear membrane breaks down. So in prophase, what we'll have, I'll draw the cell a little bit bigger this time so I can actually fit it all in, is you'll have these chromosomes becoming visible, making an X shape because they've doubled their DNA, and you'll have the nuclear membrane being broken down, and you have these centrioles, help if I use a bit of colour, you have these centrioles that have moved to opposite ends of the cell. Okay, so that's our centrioles. If we put them here, usually the centrioles in interphase are just found together like that. They're found somewhere in the cell near the nuclear membrane, okay? So the centrioles have moved to opposite ends of the cell, the DNA is condensed, and the nuclear membrane has started to break down. Next one, I proudly miss all tutorials, interphase, prophase, metaphase, okay? So the next stage is known as metaphase. Metaphase, okay? Now, metaphase is where these chromosomes that are condensed inside where the nuclear membrane used to be, these chromosomes line up in the center of the cell, and we get things called spindle fibers that go from the centrioles to the chromosomes and attach, okay? So, in metaphase, the chromosomes line up in middle of cell. I do apologise for my handwriting. I've sort of hurt my hand and I can't really hold the pen properly. So chromosomes line up in the middle of the cell, and spindle fibers attach to chromosomes. Chromosomes. Okay, so if we were to draw this, I'll do it a bit bigger this time because it looks like I'm going to have to do a new slide anyway. We've got the cell there. Our chromosomes have lined up in the middle of the cell, like so. The centrioles, which move to opposite ends of the cell in prophase, are still found at the opposite ends of the cell in metaphase, except this time spindle fibres, so spindle fibres, which are these in blue, have extended from the centrioles and attached to the chromosomes in the middle of the cell. Okay, and that's metaphase. They attach to something called the centromere. The centromere. Okay, now the centromere is sort of like the middle part of each of the chromosomes. It's a part where the spindle fibres can attach. These are known as the centromere. Centromere. Okay, so interphase, the cell is in its resting phase, DNA replication happens, ATP is made. Prophase is where the chromosomes become visible, the centrioles move to opposite ends of the cell and the nuclear membrane breaks down. And in metaphase, chromosomes line up in the middle of the cell, spindle fibres attach to the chromosomes. Okay, are we okay there so far? Am I alright to wipe the screen off? Just let me know in the chat, please. Brilliant. So let's go on to the next bit. So we've got interphase, prophase, metaphase. I proudly miss all tutorials. So we've got anaphase and telophase left to do. So if we go back to here, pick my yellow again. So I proudly miss all, we got telophase. Okay, telophase. Oh no, hang on, we haven't done anaphase. Sorry. So we got anaphase. Sorry. So anaphase. Now, what we had was the cells, the chromosomes lining up in the middle of the cell, and we had the spindle fibres that had attached in metaphase, okay, from these centrioles. Now, in anaphase, what happens is the spindle fibres that were attached to these centromeres on the chromosomes pull, okay? And when they pull, it separates the chromosomes at their middle, okay? It pulls the chromosomes to opposite ends of the cell. Now, because the cell's DNA are divided, are doubled, okay, in interphase, we have an exact copy of each chromosome, okay? So just to be clear, when DNA condenses into a chromosome, this would be considered one chromosome. But when it doubles, when the DNA doubles, there is a centromere formed, and the DNA doubles across the centromere like this. So what you have is two copies of the same chromosome. Okay? So when these are pulled apart, each side of the cell has an exact copy of each chromosome. They are identical. So in anaphase, chromosomes are pulled apart by the spindle fibres. Spindle fibres, and each chromatid, one of these is known as a chromatid once it's pulled apart. Each chromatid moves to opposite ends of the cell. So chromatid move to opposite ends of the cell. Okay, so the chromatids move to opposite ends of the cell. That is anaphase. Okay, the final stage is telophase. Okay, so in telophase, we've had these chromatids move to opposite ends of the cell. So telophase. Okay. Chromatids have moved to opposite ends of the cell. Essentially what happens is they unwind, they become stringy bits of DNA, and the nuclear envelopes form on both cells. Okay, so in telophase, if I changed the colour back, in telophase, chromatids chromatids are in opposite ends of the cell. The DNA decondenses, or the chromatids go back into stringy DNA. So chromatids unwind, and the nuclear envelope, so the nuclear envelope reforms. Okay, so essentially what we have here is we have the cell that has sort of started to do this, like so. You have the nuclear envelope, which has reformed in both cells, and you have the DNA, the chromosomes, that have decondensed to form stringy DNA again. Now what you'll notice is that these cells are still joined, there's still this middle bit, and we're at the end of mitosis. We've had I Proudly Miss all tutorials, we've had interphase, prophase, metaphase, anaphase and telophase, but we've still got one cell that is joined at the middle. Something that happens after telophase but is not part of mitosis is cytokinesis. Okay, cytokinesis. So cytokinesis is a process where the cells divide, but it is not part of mitosis. Okay, so cytokinesis is where the cell divides, but the important thing here is that it is not part of mitosis. Okay, those are the stages of mitosis. So we have the cell, which starts with an interphase where it doubles all of its DNA, it doubles its organelles, it grows and it produces a lot of ATP ready for mitosis to take place. Once the cell is big enough and it has enough DNA and it has enough organelles, what will happen is the cell will go into what's known as prophase. In prophase, the chromosomes condense and become visible. You actually get to see the DNA. The nuclear envelope breaks down and the centrioles move to opposite ends of the cell. Okay. Once that happens, the cell can then go into metaphase, where the chromosomes line up in the middle of the cell and spindle fibres attach from the centrioles. In anaphase, the spindle fibres pull on the chromosomes, separating them into two halves. They pull each chromatid to opposite ends of the cell. Then in telophase, we have the DNA which goes back into being stringy. It's no longer a chromosome. You get the nuclear envelope that reforms. You get this bibble in the bit, I can't remember what it's called, this part in the middle that starts to show where it's going to break. And then after mitosis has taken place, you get cytokinesis, where the cell actually divides. Now, the important thing here is to remember that this is in somatic cells. The cells that are formed are identical. We started with these chromosomes that you started, as I said before, and I'll draw it again just to make sure. We've got our centromere here. We've got our chromosome, which is found on the centromere, sort of as a single stick. In interphase, that DNA doubles, so this clones itself, and it's joined at this centromere, okay? When the cell is pulled apart, during anaphase, when this chromosome is pulled apart, it's pulled into two chromatids, like so. These chromatids are identical to each other. There is no difference. So through mitosis, you've got two cells that have identical DNA. There is no difference between them. They are identical. Okay? Are we okay with that thus far before I move on to meiosis? Can you just let me know in the chat, please? Any questions at all about mitosis? Brilliant. We're okay with that. Okay. So let's move on to meiosis. Okay. Now, meiosis is a little bit different, and luckily for you guys, you don't actually need to know the stages. All you need to understand is the process and what it's actually needed for. Okay. Now, while mitosis is involved in somatic cells, it's involved in the replication of body cells, meiosis is slightly different. Meiosis is involved in reproducing gametes. Okay, it's involved in making more gametes. So gametes are essentially sex cells. Okay, they are the sperm and the egg. Okay, so these are sperm and egg cells. Okay, now all the cells of your body go through mitosis, apart from the sperm in your testicles if you're a man, and apart from the egg cells in your ovaries if you are a girl, okay, or a woman. Now, this is where meiosis comes into play, okay, meiosis. Now, meiosis, unlike mitosis, is cell division in gametes. And one of the key differences in meiosis is that it results in resulting in four unidentical cells. Unidentical haploid cells. Haploid cells. Now, haploid cells, just so you know what I'm going on about, haploid cells have half the chromosomes. Okay, so your DNA comes from half your mum and half your dad. So half of your DNA comes from your mum, half of your DNA comes from your dad. This is because when your mum and dad had sex to make you, the sperm met the egg. So the sperm gave you half of your DNA, the egg gave you your other half, it joined together to form the first cell that was you. Now, The way in which these gametes are made is through meiosis. In the testicles, we have meiosis taking place, producing lots of sperm that are all haploid cells. They all have half the number of chromosomes. So in humans, that would be 23. Sperm cells have 23 chromosomes. Egg cells have 23 chromosomes, whereas other cells of your body have 46. Okay? Now, the next important thing is that they're unidentical. None of the cells made in meiosis are identical to each other. They are always different. If you got the cells of a human and you let the cells go through meiosis, you would never ever make two cells that are identical. If you think about it, this is why you look different from your brothers and sisters. If meiosis produced the same identical cells every single time, your brothers and sisters would look identical to you because you share half your mom's DNA and half your dad's DNA with your brothers and sisters. Each one of you has half of each. Now, if they were identical, you would all look the same, but you don't. You look different from your brothers and sisters. You can have small resemblances, you can look slightly the same, but you always have little differences. Okay? Now, in terms of your exams, you can often be asked, give two ways in which mitosis and meiosis are different. Okay? For example, meiosis creates 4 cells, mitosis creates 2. Meiosis creates unidentical cells, whereas mitosis creates identical cells. Meiosis makes haploid cells, whereas mitosis makes diploid cells. Those are the sort of answers you need to be giving. It's an easy couple of marks. So just to be clear, meiosis is cell division in gametes, which results in 4 unidentical haploid cells, whereas gametes are sex cells, such as the sperm and the egg. Now, another thing that you can be asked to do in terms of meiosis, okay? You can be asked to talk about how something called variation is brought about, okay? So, variation, just so we can have a definition at the top of the screen, is defined as differences among species. So, differences among species. So, for example, we have lots of variation among humans. Some have black hair, some have blonde hair, some have green eyes, some have brown eyes, some have white skin, some have brown skin. Those are all variation, okay? Those are all examples of variation. Now, the important thing is that you understand how variation is brought about in meiosis. So, if we have a look up here, so we can say variation in sexual reproduction / meiosis. I will explain these, but if we just lay them out on the line to begin with. First of all, the first way in which variations brought about in meiosis is through something called crossing over. Crossing over. Second is to do with something called random assortment. Random assortment. And thirdly, which is not really to do with meiosis, but it's to do with living things and how variations brought about, is through choice of sexual partner. You don't have to have a child with someone with blonde hair if you really don't want to. Yeah, you can choose the traits of your partner and hopefully those traits get passed on to your offspring. So the third way variations brought about in sexual reproduction is through choice of partner. Okay, so let's have a look at how we can explain these traits. So we start in meiosis with a cell, like so, and inside that cell we have the nucleus, and inside the nucleus we have what are known as homologous chromosomes. So you've got chromosomes that are from mum and chromosomes that are from dad. We say that they are homologous because they're the same size, okay? And we've also got other chromosomes from mum, but we'll say these ones are homologous because they're smaller. Add the other arm onto there. And we've got homologous chromosomes from dad, and we'll use red here. So just to be clear, homologous chromosomes are chromosomes that carry the same genes but different alleles. Okay? So we can literally say, remember I've always said in my classes that homo means the same in biology. So homologous chromosomes are chromosomes with identical genes but different alleles. So chromosomes with same genes but different alleles. Okay, but different alleles. Now the important thing is that you get homologous chromosomes from your mum and your dad. You get the same chromosome from your mum and your dad, but they have different alleles. So you have 46 chromosomes. You've got 46 chromosomes in your body, but they are arranged into 23 homologous pairs. Okay? So, in meiosis, we start with a cell that has its homologous chromosomes about in the cell. Okay, they get arranged so that they are next to each other. Now, the first stage in meiosis by which variation is brought about is through crossing over. Okay, now in crossing over, what happens is we get a part of each homologous chromosome swapping. So, save drawing the whole cell. I'm just going to draw the nucleus for now so you can see where I'm coming from. So just to be clear, this is just the nucleus. What we get is we have this green chromosome swapping a part of its arm with the orange one or the yellow one or whatever colour it is, okay, like so. And we get this orange one swapping part of its arm with the green one. Okay? Again, that happens with the blue and the red one. So in homologous chromosomes, we get this known as crossing over. Arms of the DNA is swapped with homologous chromosomes, like so. So if you look now, no two of these chromosomes are the same. We've got a green one with an orange arm, and we've got an orange one with a green arm. We've got a blue one with a red arm, and we've got a red one with a blue arm. Okay? So we've got cells that are all totally different. We're going to create cells that are totally different. This is known as crossing over. Arms of each homologous chromosome are swapped at the middle. Okay, that's a very difficult concept to understand. That's something that people get stuck with. So if we say this is crossing over, can I just ask in the live chat that you let me know if there's something you don't get, please? Do you understand that about crossing over? That's an excellent question, Callum, about identical twins. So with identical twins, meiosis has already taken place. Identical twins are formed when you actually get the embryo splitting. Okay, I will show you that at the end of meiosis. That's an excellent question. But are you okay with this idea of crossing over people in the chat. Just let me know. Is there anything you want me to explain again? Brilliant. Okay. So in crossing over, we get these homologous pairs swapping arms. Now, the next part that brings about variation is this random assortment. Now, what that means is that when the cell divides in the first round, so there is 2 rounds of cell division, sometimes you'll get them pulled one way and other times you'll get them pulled another way. So for this case, the spindle fibres, much like in mitosis, this time you might get this one pulled this way with the green arm at the bottom, and you'll get the green one with the orange arm at the bottom pulled this way, and you'll get, let's say this time you have this red one pulled to the left side with the blue arm at the bottom, and we'll have the blue one on this side with the red arm. Okay, so no matter how many times you run this, you'll never get the same chromosomes pulled to the same side. This is known as random assortment. The chromosomes will be pulled to different sides of the cell every time you do it. If you do identical cells every single time, they will always be pulled to opposite ends of the cell. Okay, this is known as random assortment. Okay, so they're pulled to opposite ends of the cell. Now, what we've got is this would then divide into 2 cells. Each cell, so I'm running out of room to draw if I'm honest. So I wonder if I can, okay, perfect. So if we, oh, it won't let me. Can I draw on there? No. Okay, so never mind, we'll just move over to the side. Oh God, it's all gone Pete Tong. Wow. Oh dear Lord, what's happening now? Okay. It was all going so well. Okay, we'll have to work with it like so. So you've got your random assortment where they're pulled to there. What that results in is you end up with a cell that has the part green, part yellow, and you have a cell. So we've got a cell that has a all green with a part of yellow, and we have a cell that has all red with a part of blue. It'd help if I'd left the red. So all red with a part of blue, and then we've got a cell that has a part of red with the rest blue, and we've got a cell that's all yellow with a part of green. So all yellow with a part of green. So we've divided the cell up. What happens then is that the cell goes through another round of division, except this time these sister chromatids are pulled apart exactly like we had in mitosis. So when these sister chromatids are pulled apart, what we are left with is 4 cells. 1, 2, 3, 4. So this first cell will have 1 cell that has all green and 1 cell that has a bit of red and a bit of blue. So half red, half blue. We'll have a cell that is half green and half orange, half green, half orange. We have a cell that has all red in there, like so. We have a cell that has half orange and half green. We have a cell that is all blue. And we have a cell that has all orange and half blue, half red. Now, if you look, no two of those cells are the same. We've gone through 2 rounds of division where we've started with the homologous chromosomes there. We have crossing over take place where some of each arm of each chromosome is swapped. We then have the cells pulled to, the chromosomes pulled to opposite ends of the cell. The cell divides and it has random assortment where these chromosomes are pulled to random sides of the cell. These cells then go through another round of division to form these 4 cells. All of the cells are haploid, they have half the amount of DNA, and each one of them is different. No 2 of them have the same type of chromatids, the same type of DNA. Okay, this is where we get these 4 haploid unidentical cells. If these were to go on and fertilise an egg, you would get a different combination of alleles every time. Therefore, that's why you don't look like your brothers and sisters. So, for example, this sperm cell could have made you, whereas this one could have made your brother. This one could have made your sister. And there's these slight differences that cause differences. Now, Callum asked an excellent question about, well, what happens with a identical twin. So with an identical twin, what you'll get is you'll get this sperm fertilising the egg becomes an embryo. An embryo is just a ball of cells. So you get the egg, sperm that fertilises the egg to make the first cell that makes you. That then divides into 2, which then divides into 4, which then divides into 8, into 16, and so on and so forth. But at some point of this stage, with identical twins, this embryo will break into 2, like so. And those 2 embryos will keep developing and form 2 identical twins that have the same DNA. However, this is because the embryo splits. This is nothing to do with meiosis. Okay, so, Courage, I can see your name, yes. So, I think for today, that will have to do, okay, because I've got things I need to go on and do. I only intended for this to be an hour, but because I had trouble with the setup. What I will do is I'll send out an email. Okay, I will send out an email later today and I'll explain when we're going to do the next session. I may do it later today, it may be tomorrow. Okay, I have seen that somebody asked about protein synthesis, DNA transcription and translation, Casey. I can go over that tomorrow. That will be the first thing that I go over. Okay, now what I will say is if you drop me an email of anything you want me to go over tomorrow when I do another one, please let me know. I am intending to do a GCSE one as well. I'm going to look at doing a GCSE live stream. So if you have got brothers and sisters who want to come on, that's fine. I don't mind them dropping on and having a look at what I've got to do. Okay, but I hope that has been of some help. If you like this sort of learning, I don't mind doing that when you go on study leave as well. I don't mind bringing my laptop into school and doing this for the hour that you're supposed to have me. Okay, but I hope that's been useful. Please send me any emails of any questions you want me to go over, and I hope you enjoy the rest of your sunny Tuesday. I'll send out an email. I'll probably do another one of these tomorrow. Okay? Anyway, I'm out. See you later.